Imagine you're really hungry and you eat a burger. You don't feel the burger turning into an energy. There is no buzzing or sparks in your stomach, but inside your cells, something amazing is happening, and that is when enzymes came in. Enzymes are special proteins in our body that speed up chemical reaction. They make reaction happen even faster and easier. It works by lowering the activation energy and they can be reused in many times. Enzymes work on a specific substance called substrate. This is where the substrate fits and it is called the active sites. The enzyme substrate interaction model is also known as induced fit model. Induced fit model is a model of enzyme substrate interaction where the binding of the substrate causes the change in shape of the enzyme active site to achieve a tighter, more precise fit. It also influenced by factors such as temperature, pH level, substrate concentration, enzyme inhibitor, and activators. The mechanism of enzyme action is where the substrate binds to the active site. The enzyme substrate complex is formed. Chemical reaction occurs. Enzyme releases product. Enzyme remains unchanged and reusable. This is how the enzymes work. As it grabs the substrate, they form a temporary connection. Then the reaction happens quickly and the products came out. And then the enzyme is free again to repeat the process. Without the enzymes, many reactions in our body would be too slow for life to continue. They are needed for digestion, breathing, healing, and energy production. There are also factors affecting enzyme activity, such as environmental factors, which is the temperature. If it is too low, then the reactions will be slow too. If it is too high, then the natering will may happen. For the pH level, each enzyme has an optimum pH ranges to 6 to 8 pH level. For the substrate concentration, more substrate increases the reaction rate until saturation, and enzyme concentration, meaning more enzyme is faster rate. For the inorganic substances, these are essential for the function of the enzymes that are known as cofactors. These are the typically metal ions that can help stabilize the enzymes structure and directly participate in the catalytic process. There are also two inhibitors, which are the competitive and the non-competitive. How can we use the enzymes in real life or everyday life? For our body, we have the enzyme called amylase, where we can find in our saliva, and it breaks down starch when we eat. And also, lactase help us digest milk. Lipase and protease break down fats and proteins. In foods and drink, enzymes make cheese, bread, or yogurt. Brewing companies use enzymes to make beer and juice clearer. For cleaning products, laundry detergents use enzymes to remove stains like oil using the lipase, protein using the protease, and starch using the amylase enzyme. For, medic- for medicine and science, enzyme help in blood tests, disease detection, and DNA tests. To sum it all up, enzymes are proteins that speed up reactions. They work with a specific substrate in active site. They are used in digestion, cleaning, food production, and biotechnology. And without enzyme, the body reactions would be too slow to keep us alive. Understanding enzymes makes biology feel more connected to everyday life. That's all, and goodbye.